So this is Emily Perry again, and in this next presentation we're going to be talking about variation data in Ensembl. So I'm going to talk about how we classify variants, where we get the data from. We're going to have a look at the gene location and variant tab to see how you can find variants and information about them in Ensembl. And then finally we'll look at the variant effect predictor, or VEP. So we classify variants according to their size. We have short variants, such as single nucleotide polymorphisms, and small insertions and deletions. And we also have structural variants. These would be large-scale copy number variations, inversions, translocations. We don't have variation data for all the genomes in Ensembl, but for quite a large number of them. And if you're working with non-vertebrates, we also have variation data for a number of species. A lot of the species actually, instead of having variation data, we just have strains. So the Ensembl variation process consists of our import of data, quality control, adding linked data, and then carrying out analyses. So the import of data, we use publicly available archives such as DGVA, DBSNP and EVA. And we have some pages that list all the different sources we have. So for species like human, we have many millions of variants. We also use somatic mutation resources, so we get data from sources like Cosmic for human. Because these are somatic mutations, they've occurred in the tissues, there's no allele frequency or population information for these variants. So then we do quality control, so we map the variants to the reference assembly. Sometimes this means remapping variants, for example NOMAD variants from GRCH37 onto GRCH38. We check the alleles, we make sure that at least one of the alleles matches the reference assembly. We check for ambiguity codes and we exclude any suspect variants, so things that perhaps don't match or map to multiple locations in the genome. We then add linked data, so there is absolutely tons of extra data out there from other databases that we can add. So as well as we might get the variant itself from DBSNP, but then perhaps we would import phenotype related data from the GWAS catalogue, allele frequencies from 1000 Genomes or NOMAD, or publication data from Europe PMC. The population frequencies we use in order of kind of how new they are, how old they are, starting with HapMap, which is worldwide genotyping. We then use 1000 Genomes data, which is kind of an update on HapMap, and this is healthy whole genome sequencing from populations all around the world. NOMAD is mixed disease and healthy whole genomes and exomes. These are imported from NOMAD. It was made by pulling together data from huge numbers of sequencing cohorts, which means it has a mixture of disease and healthy, has a mixture of whole genome and whole exomes, but it is skewed towards the most studied populations. We also have UK10K, which is UK-wide disease exomes, and TopMed, which is disease whole genome sequencing. So the analysis we do is then comparing the variants to some of the data that we have in other parts of the database. So we calculate variant consequences and protein function prediction by comparing to our known genes. We carry out linkage disequilibrium analysis by comparing the variants to each other, where we have haplotypes from sources such as 1000 Genomes. We also have carry out analysis of variant conservation to identify things like ancestral alleles. So the variant consequences depend on where within a gene the variants fall. So we'll classify things on whether they fall in a coding region or not, whether they change the amino acid sequence. We use as standard sequence ontology consequence terms, which is a standard set of terms. There's also a consistent colour code that you'll see throughout Ensembl for labelling variants according to these consequence terms. So variants will be labelled with their most severe consequence. And severe consequence we tend to show in kind of danger colours like reds, oranges, pinks. And sort of less severe variants we show in sort of safe colours like greens and blues. We predict the effect of missense variants, so we use lots of different algorithms to give scores or ranks for changes in amino acid sequence. So this would be only for missense variants. These scores are based on things like how well conserved the protein is, the chemical change in the amino acid. So going from a positively charged amino acid to a negatively charged amino acid would very likely affect the function of the protein. It also takes into account 3D structure and domains. It's important to note that all of these predictions are predictions. They're not facts. They've not been experimentally validated. You can't 100% rely on these for the interpretation of variants. They're more as a sort of guideline for future experiments. So some of the algorithms that we can give you predictions for are SIFT, Polyphen2, CAD, Revel, METAL-R, and Mutation Assessor. And as you can see, all of these different methods have different sets of scoring, different thresholds that they set, but they do come with these predictions, so you don't have to remember what the scores all mean. You can just find the prediction. Wherever you see them, they'll be in a sort of coloured box in Ensembl, and you can mouse over it and it will tell you the terminology, such as tolerated or benign or likely deleterious. I just want to make a point about reference alleles, and this is related to the way that that the genome was created. I mentioned in the first presentation about how the genome was made up of chunks of sequence from real people. This means that if you are looking at a genomic region, then the sequence in that genomic region actually comes from a person. So if we consider this particular base position here, this highlighted T is what this individual had at this locus. However, this T might actually be a low frequency or even a private allele that was only found in that individual. It might be that the other allele is the one that's found in all primates, so it can be labelled the ancestral allele. And it might be that this allele in the reference genome is the one that causes the disease susceptibility. But because this allele, this T, is in the contig, it's in the reference genome, that makes it the reference allele. The reference allele always refers to whatever is in the reference genome. It has no relation to whether it is rare or frequent, or whether it's disease causing, or whether it's ancestral. And the standard nomenclature is to quote as reference/alternative. So in this case it would be T/G. It's also important to remember about allele strand. Variants are not single allele, single strand. They always affect both strands, because the DNA repair machinery will make sure that the two strands always match. So if you have a TG variant, you know that actually that's TG on the forward strand, and if you looked at the reverse strand you would see that it's actually AC. Certainly if we're talking about an exon which is on the reverse strand, we would describe the variant in that exon as being AC. However, when we're talking about the variant itself, independent of the gene or the exon, we will always refer to the forward strand alleles. And this is a convention not just used by Ensembl, but many large-scale sequencing projects such as Nomad and 1000 Genomes. Once you start looking in the literature and in databases which are curated directly from the literature, you may find that they don't refer to a particular allele. They will do whichever they prefer. And this could be because they're talking about a gene on the reverse strand, or it could be the method of sequencing that they used. They may not tell you whether they're talking about the forward or reverse strand, so this is something to be quite aware of when you're you're reading about variants perhaps associated with a phenotype. If you had an A/T and you were told that T was associated with the phenotype, unless you know for certain that that T is referring to the forward strand, you don't, or the reverse strand, you don't actually know which allele is associated with the phenotype. So this is the process. So you can find the data on the website through the Variant Effect Predictor and through Biomart, and you can get the data programmatically. So we're going to have a look at a gene, AGT. We're going to find variants in the gene. We're then going to look at the region and find variants in the region. And then finally we'll look at a variant, RS699, and find out more information about it. So I'm going to jump out of the presentation and I'm going to go to the Ensembl homepage. There I'm going to search for my gene, AGT, and I'll go to the Gene tab. So as I showed you in the presentation earlier, you can look at sequence and you can see variation data on the sequence. So I'll just click on sequence here. Now because I added the sequence data earlier, it's showing up, or it will show up when it's loaded. So the variants are shown as these coloured highlights on the sequence, and you can see as I put my mouse over this, so they show up as IUPAC ambiguity codes. The colours match the colours in this legend at the top, but if I put my mouse over it, you can see that this R ambiguity code actually stands for G/A. Now what's quite interesting is if I click on this, and now I get a link into the database describing this variant, and in this link for the same variant I just hovered over before, you can see it describes it as C/T and tells you that's the forward strand. So when I put my mouse over it, the alleles I was getting were actually the alleles in this sequence, and this header in the FASTA with the -1 at the end indicates that the sequence I'm looking at is reverse strand. So when I hover over, I get the alleles in the reverse strand because I'm looking at reverse strand sequence, but when I click on it, I get the alleles on the forward strand. So I can explore variation data in a gene in a table as well. If I click on variant table here, this gives me a table listing all the variants found within this gene. So I can click on the identifiers and explore them more. I've got these evidence statuses. So if I hover over them, I can see what they mean. And these give me an idea of how likely it is that this variant is real. If it's got a lot of evidence status, particularly if it's got things like multiple submitters or frequency data, this tends to indicate to me that this is a real variant. But if it didn't have any evidence status, it may just be from a single submitter and may not be real. It gives me the consequence in the different transcripts, so you can see that we get the transcripts repeated in the list. And I can choose to filter this table to only see the variants I'm interested in. So if I click on consequences, and let's say I go for protein truncating and missense, and then hit apply, this then narrows the table down for me. So now I can only see missense and protein truncating variants such as this frame shift, or maybe stop gained and things like that elsewhere. As you can see, because I'm looking mostly at missense variants, I have things like amino acid substitutions listed and amino acid coordinates, and I also have scores from all these different methods of predicting missense effects. As you can see, they don't always agree with each other. In the case of this variant here, CAD predicts this to be a perfectly likely benign variant, but most of the other prediction methods are telling me it's likely to affect the protein. So they don't always agree with each other. If you're colourblind and you can't see these reds and greens and yellows, just put your mouse over and you'll get the phrasing. We can also see structural variants in the gene. So if I click on structural variants here, I get a graphic showing me the structural variants against the gene and also a table listing them. If I want to know the phenotypes that have been linked to this gene, I can go to that in the menu at the side. So here I can see that this gene has been associated with hypertension, but I also get all the phenotypes associated with variants that fall within the gene. And further down, I get all the phenotypes associated with orthologs of the gene as well. I'm going to go into the transcript tab now because there is an interesting page in the transcript tab only. So I'm just going to click on this transcript tab at the top here. And I'm going to go to the haplotypes page. And the haplotypes page features data from 1,000 genomes. So the great thing about 1,000 genomes was that the data was phased. So we're able to calculate the actual sequences for different proteins that individuals within 1,000 genomes had. And so we can get frequencies for these different haplotypes, and we can see that, for example, this haplotype with 2 different variants found within the same protein was found in 507 people within 1,000 genomes. So I click on this haplotype, I get information about it, so I can see its frequency across the different 1,000 genomes populations, and I can hover over the names to see what they are. I can see the resultant sequence. So here's my first variant and my second variant. So there's an alignment there, and then there's a sequence, the new protein with both of the amino acid changes. And I can see which samples in 1,000 genomes had this particular haplotype. So if I wanted to then order a cell line with this particular haplotype, I could do. So I'm going to go up to the top, and I'm just going to hop to the location tab, and I'm just going to open up the configure menu. And if I go into variation, you can see there's all kinds of variation data that I can add to a genomic region. So I've got sort of different sources. I can put 1,000 genomes data on by population. I can put data on by phenotypes and sources. So I can plot different kinds of data to do with variation against the genome. What I'm going to do now is actually search for a variant. So I'm going to search for RS699, and I'm going to click through, and this will now take me to the variation tab. So we've had a look at the gene tab and the transcript tab, but this is the variation tab, and I'm afraid my icons are not appearing, which is a real shame. These are all supposed to be pretty icons, but if I put my mouse over them, you can see what they're supposed to be. So I have some information about this variant. I can see that its most severe consequence is its missense. I can see its alleles, its minor allele frequency. But if I want to explore more, I need to use these buttons here. So I'm going to click on this button, which would normally have a picture of a gene on it, and this will tell me the consequences of the variant on things like genes and regulatory features. So here we can see that only one gene is actually hit by this variant. It's missense, and we can see its amino acid change and its codon change, and where within the gene the variant falls. I also get gene expression correlation. So this is data from GTEx, and this tells me all genes that have their expression increased or decreased by this variant. So we have a number of different tissues listed here. If the variant hit regulatory features or motif features, these would be listed here as well. Another way I can look at how it affects the gene is in the 3D protein model. So I'm going to click on 3D protein model in the menu here. And this gives me a protein structure from PDB. It's again in light mode, just as we saw with the in the transcript tab. And if I spin this around, you can see that our variant of interest is highlighted here in red. So we can see where it falls within the protein structure. We could use this to identify if it falls in something like an active site. We can find out more about the variant, so its population frequencies. So I'm going to go to population genetics. And here we can see the frequency in 1,000 genomes populations, but if I scroll down the page I can also see the frequency in Nomad exomes and genomes and TopMed and other sources. So you've got these little pie charts which I can expand, so let's expand the African one, and you can see the different subpopulations within the African group. So if I put my mouse over again, I can see what these 3-letter codes stand for, and I've got more detailed data further down, so I've got not just my allele frequencies but my genotype frequencies. So this allele is variant is quite skewed across the populations. You see that the A allele is most common in the European population, but in all the other populations the G allele, particularly the African population, the G allele is much more common. If I go into phenotype data, I can see that this variant has been linked to a number of different phenotypes, including coronary artery disease, hypertension, mean arterial pressure, so quite a few different conditions. So we've got the sort of name of the phenotypes. These mapped terms are a phenotype ontology, allowing us to link together terms that mean the same thing, and you can see clinical significance from ClinVar and some statistics as well as to how strong this link is. I can also see the gene and find out if there are any phenotypes linked to the gene. If I click on this coronary artery disease, so it takes me to a page that lists all the other variants which have been and genes which have been linked to coronary artery disease. So I can see this list of variant IDs, the genes that are linked to them and their locations in the genome. So now I'm going to go back into my presentation and I'm going to talk about the variant effect predictor. So the variant effect predictor is a tool to predict and annotate the functional consequences of variants. So I said as part of our data pipeline, we analyse the variants to see what genes they fall in and find out information about them. And link to other data. If you've got a list of your own variation data, maybe you've done some variant calling, you can actually run the VEP and do similar kind of analyses on your list of variants. So we take input into the VEP in a number of possible different kinds of formats. So we can use variant coordinates, we can use HGBS notation, which expresses the position of the variant in terms of a gene, a transcript or a protein. We can use VCF, which would be your standard output from any kind of whole genome variant calling. We can use a list of variant IDs, or we can use the new SPEEDY format from dbSNP. So we will predict the consequences of variants on known ensemble genes. And you can actually use ensemble genes, or if you prefer, you can use RefSeq genes. You can use both ensemble and RefSeq. We will tell you if your variant overlaps regulatory regions, which come from data from ENCODE Blueprint and Roadmap Epigenomics. And you can limit this to regulatory regions that are active in specific cell types. We'll also give you the pathogenicity predictors, and if your variants are known, it will find out, it will find the variant and it will say, seen this one before, this is its ID, and it will tell you if it's been linked with a phenotype or a disease, and give you its clinical significance. You can also get plugins which add extra functionality to the VEP. They can extend, filter or manipulate the output, and they can make use of external data or code. Some of them are available on the web tool, and all of them are available with the script. So the VEP works, there is a web interface, which is what we'll use today, but there's also a standalone Perl script that you can just run on your computer, and you can access it through our REST API. It's not just species which have variation data that work with the VEP. Any species within Ensembl and any division of Ensembl, you can run the VEP on it using our files and our data. But if you're working with a species not an Ensembl, you can still use the VEP. So one of the ways that VEP can work much more quickly is by using an offline cache. So this is basically a load of files and data which contain everything the VEP needs to run. The advantage of the caches, as well as speeding it up, is they help keep your data private. And there are pre-built caches available for Ensembl species, but if you're working with something that's not in Ensembl, you can make your own if you've got GTF and FASTA files. So even if you're working with something like a T-Rex, you can still run the VEP with it. There is a paper on the variant effect predictor by Will McLaren from 2016 if you would like to learn more about it, and if you use it, please do cite that paper. But I'm going to do a quick demo using the VEP, looking at a set of 4 variants to show what genes they hit and what effect they have on them. So I'm going to escape my presentation here. I'm going to select these data. I'm going to go back to Ensembl, and I'm going to hit VEP up here in the blue bar. And I'm going to scroll down. I've got options for documentation and information about the different ways to access VEP, but I'm just going to hit Launch VEP. And I get this jobs page. If you've run VEP before, you might get a table listing your previous jobs. So this is just my new job. So I can give my data a name. So I'm going to call it demo. And then I'm going to paste in my data. So I'll just paste in the data I had before. You can also upload a file, so you can choose it from your machine. Or if the data's online somewhere, you could just put the location of the file in here. And I can choose to use different transcript databases. I'm going to stick with Ensembl GenCode transcripts. Now I've got a bunch of different options, so I'm just going to expand these and show you what's there. So if I go into identifiers, you can see I'm going to see my gene symbol and the version of the transcript. I'm going to add HGVS because I think that's quite useful. I'm going to open up variants and frequency data, so you can see this is going to find us co-located variants, and it's going to get us some frequency from 1,000 genomes. I can get additional data, so things like the biotype of the transcript support level, all of these bits of information that I might want to use to filter down to a preferred transcript. And I'm going to tick phenotypes to find out if my variants or genes have already been associated with phenotypes. I'll also pick protein domains because that can get me the protein structures. It's going to get me SIFT and polyphen scores, but I could add other kinds of scores such as CAD or Condell and things like that, and also splicing predictions. I can at this point filter my data, so I can choose to, for example, if I go into advanced filtering, I can choose a threshold, a frequency threshold, and I can choose my favourite population for that, but I'm going to go with no filtering because I've only put 4 variants in. You can also change the buffer size, which may change the speed of the VEP. So I'm going to hit run. And now it shows me my job in this table. So it's currently saying it's queued, and it's going to refresh for me every 10 seconds until it's done. I can save the job to my account. So if I go up here, you can see I've got login/register. I can set up an Ensembl account, which is free, and nobody ever sends me any spam. So I could save it to that account if I wanted to. So now it's telling me my job is done. So I'm going to hit view results. And I can see that my 4 variants have given me lots and lots of consequences because they hit multiple genes and multiple transcripts of those genes. One of them is novel and 3 of them are existing. I've overlapped 2 genes, 7 transcripts and 1 regulatory feature. As I scroll down, I get the detailed information. So I've got a list of all my variants. It has changed the order of them. And information about the consequences that they have. So if I scroll across the table, I can see information about the gene that they've hit. So I can get the gene symbol and the ensemble gene ID. Also the transcript identifier. Because I ticked HGVS, I've got this HGVS notation. So this one here is a synonymous variant, and as I scroll across you can see I've got HGVS-P and HGVS-C. I can see where in the protein it fell, so it's only got 1 amino acid because it's synonymous, but I can still see whether the codon has changed. I can see that this is an existing variant, so this variant was already known in the database. And as I scroll across, so you can see it was found in some protein domains. If I scroll down here, further across, I've got one here that hits a transcription factor binding motif, so I can see where within that motif the variant was found, and the change in the motif score. And down at this far end, I've got associated phenotypes. So I can see that this particular variant was linked to obesity in the GWAS catalogue. But if I scroll down, I also get links from the gene. So this gene has been associated with this very long phrase by OrphanNet. So I've got lots of information in this table. I can filter the table to show only the data I'm interested in. So let's say I only want to see missense variants. I can add this filter. And now the only variants in my table are listed as consequence missense. And if I scroll across, you can see I've got things like Sifton polyphen scores, amino acid changes, and other data like that. So that's the variant effect predictor, and that's everything I wanted to say about variation data in Ensembl. And there are some exercises to do looking both at the variation data in the browser and at using the